Welcome everyone to What's New in MPMI, the virtual seminar series of the MPMI Journal. I should introduce myself. I'm Jean Harris. I am the host and organizer of the What's New in MPMI seminar series, where we select papers from the journal to bring to you for a chance to interact with, engage with the authors, and learn something new. So while everyone's coming into the room, I'd like to invite you to just type into the chat where you're from. We are a very international community. We have lots of people joining us. So just type into the chat your name and where you're coming from, and then we'll be able to welcome you all. Okay, we have Karen from the Netherlands here. Who else is coming? Ben Petre from France, welcome. Ashley from Fargo, North Dakota, from the Friesen Lab. Orielan, oh, it's going too fast. Poisson Dernier, sliding too fast, from France. Robert West from Houston. Yvonne Mukuka from Australia. That's middle of the night, I think, for you. Kelly from Colombia. Paul Covey from Colorado. Xiao Xuan from China. Elena from Croatia. Dilip Shah. Lucia Vignale from Uruguay, Marion from Switzerland, Matias from Italy, Juan Jose from Mexico, Enas Elbalad from Egypt, Serkan Pazarlar from Denmark, Copenhagen, the UA from Switzerland. We have people from everywhere. Chloe Ramirez from Moscow, Idaho. I'm going to have to unfortunately miss some people, but I hope you were all reading that. Franz Lee Flores from Guatemala, Minu Singar from South Dakota, and Adam Steinbrenner from Seattle. So welcome everyone. It's exciting to see you all here. For those of you who just came in, this is What's New in MPMI, the seminar series of the MPMI Journal. We are an international community, so this is a chance for us to meet everyone from around the world and talk about things that interest us. So plants live all around the world, microbes live all around the world, and so does our community. I'm Jean Harris. I'm the host and organizer of this seminar series. And one reason I started this a number of years ago was at the beginning of the pandemic. And we felt a really strong need to connect. So many things had been canceled. We felt a strong need to connect with people all around the world. And also, I had a real desire to promote inclusion, to connect our community, but also to welcome people to the community, to these really top-notch seminars, to people who might never have attended one in an international conference. And this gives everyone a chance to be in the room while we highlight exciting research from the MPMI Journal. And we also try to highlight early career scientists, as we are doing today. Yingpeng Xie is our speaker today. And he wrote, co-authored this article with Libo Shan, who is also with us, Ping He and Chao Zhang, part of the research group, who came together to create a wonderful invited review, one of our distinguished HA Floor reviews that MPMI has started under the editor-in-chief, Tim Friesen. This is an exciting and important series of reviews on key topics in plant microbe interactions. And one thing this seminar does, is gives everyone a chance to interact directly with the authors outside your screen or the pages of the journal, to have a chance to hear them talk about the research and the ideas, the scientific ideas that really excite and motivate them, and to give you a chance to ask questions. So we introduced ourselves in the chat, and I hope that everyone will read and see all the people who are coming from all around the world to join us. But when you ask questions, we will not have microphones on, but you will type your questions into the Q&A box that you see at the bottom of your screen. And at the end of the seminar, I will read them and they will be answered. So mostly in order. So It's a great pleasure. I'm not going to give an extensive introduction to Yingpeng Xie, but just to say that he is currently doing a postdoc with Li Boshan and Peng Ke at the University of Michigan, as you can see from your slide, and recently moved here from Texas A&M, and previously did their PhD in Hong Kong and undergraduate in China. So Yingpeng has also had a very international career path, as many of us have. So welcome, and I'm going to turn it over to you, Yingpeng, to tell us about unlocking nature's defense, plant pattern recognition receptors as guardians against pathogenic threats. Thank you.
Thank you, Dr. Harris, for introduction. So hello, everyone. So it's a great honor to share my topic with you today. I'm a postdoc in the Department of MCDB at the University of Michigan. And my working focus is on plant innate immunity, particularly on the immune pathways activated by the cell surface receptors. So today I will be discussing unlocking nature's defense plant partner recognition receptors as guardians against pathogen threat. So why should we study plant disease and immunity? So the answer is quite straightforward. Global crop losses and associated economic damage are prevalent. So each year, up to 40% of global crop production is lost due to damage, due to other pests and diseases. And at the same time, climate change makes these issues even worse, right, affecting our daily lives. So healthy plants with robust immune system are more likely to survive in the stressful environments created by climate change. So understanding plant disease and the mechanisms by which plants defend themselves is crucial. So let's start it with some background on our understanding of plant immunity. So the concept of plant pathogen emerged in the 19th century. So building on the foundation of its inheritance and genetics, so Flor proposed the groundbreaking gene-for-gene hypothesis in 1940s. And in the 1960s and 70s, researchers just discovered a diverse array of microbe-associated products, which termed general elicitors, which could stimulate immune response in plants, including some phytoalexins production at the species level. So the term elicitor was proposed in 1984, at the same time as cloning of the first AVR gene, AVRA. So the 1990s saw a surge in R gene cloning, so further enhancing our understanding of plant immune mechanisms. And in early 2000s, Sosen and colleagues unveiled remarkable similarity between the innate immunity in plant and animals. So this finding explained why plants recognize general elicitors and introduced the term PAMP, which refers to the pathogen-associated molecular patterns to describe these plants, to describe this recognition. So co-occurrently, there were also the increase in the cloning of PRRs. And one of the latest breakthrough in plant immunity is the discovery of the nucleotide binding and leucine-rich repeat, which referred to as the NIR resistors. And upon activation, the resistance proteins can oligomerize to form the resistance, which is a crucial component of an effective immune responses. So let's begin with the gene-for-gene hypothesis. So this theory was first introduced by Flor in the 1940s, emerged from his study on Ross disease, even before any genes were cloned. So this theory proposed that in plant disease resistance, it is governed by specific interaction between a resistant gene in plant and an avirulent gene is pathogen. So this interaction, known as AVR, and These genes are highly specific and occurred at the level of individual cultivars and pathogen strains. So this work revolutionized our understanding of plant pathogen interactions and ultimately led to the cloning of resistance genes. So after 4 decades of the gene for gene hypothesis proposed, the two cell membrane localized resistance proteins were identified. So in tomatoes, a transposon tagging technique reveals that CF9 plays a key role in recognizing the AVR9 effector protein from a fungi pathogen. So the mutant plants lacking the CF9, as shown here with this arrow, exhibited a survival response to the hypersensitive response, typically characterized by some rapid cell death triggered by the AVR9 protein. And similarly, in rice, XA21 protein was identified as another membrane localized resistance protein. This protein can recognize bacterial pathogens such as Pseudomonas, and in XA21 transgenic rice plants, it displays significant higher level of resistance against Pseudomonas infection compared to the wild-type plants. So identification of these two proteins introduced the concept of race-specific resistance against plant pathogen. And initially, both CF9 and XA21 are thought to be similar membrane-located resistant protein. However, after the later research classified CF9 as a receptor-like protein due to its lack of kinase domain, while X21 was identified as a receptor-like protein. X21 was identified as a receptor-like kinase due to its kinase. And both of these two proteins is critical for signaling with the plant immune system. And in 2000, a protein called FLS2 was identified in Receptors. So this membrane-located protein can recognize a small flag22 unit from bacterial flagellate. Unlike our gene, FLS2 serve as a general receptor for recognizing pathogens. So FLS2 is a leucine-rich repeat protein and contains 28 ARR repeats in its extracellular domain. So mutation in this FLS2, such as the loss of function mutant FLS2-2-17, lead to the increased susceptibility to Pseudomonas D3000 infection. This underscoring the critical role of FLS2 functions as a player in plant immune defenses. So the discovery of FLS2 is important because it is a key component for plants' first line of defense against bacterial infection. And this mechanism enables plants to detect and mount a defense response even against some unfamiliar pathogens. So FLS2 was first cloned PR in plants, and which recognize a general bacterial component rather than the race-specific molecules. And this discovery also spurs the concept of PAMP and PMP triggered immunity, also known as PTI. So following the emergence of PAMP concept, numerous studies reveal strikingly similarity between the molecular organization of animal and the plant immune system. Like animals, plants have evolved the ability to recognize PAMPs from diverse microbial organisms and absent in potential host plants. So notably, researchers also discovered plant receptor resembling mammalian toll-like receptors, as well as a conserved MAPK signaling cascade, and also the production of some antimicrobial peptides here. So what is pattern recognized receptors, aka PRR? So PRR consists of two main types of protein located on the plasma membrane. The first type is receptor-like kinase, also known as RK. This consists of three parts: an extracellular domain for the sensor on the outside, and a transmembrane domain which anchored it in the cell membrane, and also a kinase domain which transmits signals inside the cell. And the second type is the receptor-like protein, RP. This protein lacks the kinase domain and cannot directly transmit signals, but still play a key role in recognizing the external signals. So when a plant encounters a pathogen, it detects molecules called PAMP and also some self-derived molecules from DAMP, and I will introduce this later in this talk. So these molecules can bind to the extracellular domain of the these PRRs and triggering plants' immune responses. So these two types of RK and RP are known as PR. To illustrate the significance, the genome of Receptors is predicted to contain over 600 RKs and 57 RPs, while the rice has more than 1,000 RKs and 90 RPs. And the crucial role in plant defense is evident. And the PR can recognize different types of molecules based on their extracellular domain. So, for example, the RRR type receptors like the FLS2 receptors can detect microbial proteins or peptides, and other types of receptors such as the lysine motif receptor and EGF type receptors and lectin type receptors can recognize microbial sugars. So plants could perceive both external and internal signals to regulate diverse biological process. So PRR detect external signals from virus pathogens as well as molecules created by plant itself. So to date, researchers have identified over 60 immune-related PRs with no elicitators across various plant species. So PRs also perceive signals from not only perceive signals from bacteria, but also signals from fungi and myces, and some self-derived molecules, the parasite plants, or even some herbivores. So one of the most recent identified PRR is Peru, and this RK was found in potato and can recognize a PEP13 fragment from the myces. So when we discuss receptors in plant immunity, a crucial question is, how is an extracellular input was transduced into an intracellular output, right? So for PRs, this process often involves dimerization with another receptor-like kinase, commonly referred to as a co-receptor. So upon recognition elicitor from pathogens, the phosphorylation of these components is essential for activating the immune signaling cascades. And remarkably, phosphorylation occurs very quick, typically within seconds after binding to the elicitor. So one of the most well-studied PRR complex is Flectin-2, FLS2, BAC1 system. And this image shows the crystal structure of the active domains of FLS2, BAC1, and Flectin-2. So basically, Flectin-2 binds to 14 ARRs to the concave surface of FLS2. So Flectin-2 bind FLS2 active domain directly interact with the BAC1 active domain, and the C-terminal region of the FLS2 bond Flectin-2 work as the molecular glue, and it could stabilize the FLS2 BAC1 dimerization. And this process is both ligand and receptor mediated. This means this immune complex is very precisely regulated. Right? So downstream PR complex are receptor-like cytoplasmic kinase, IRCK here. So IRCK predominantly contain serine or threonine kinase domain. So ILCK can be classified into 17 subfamilies, with the most notable example being the big one, PBS1, and the PBLs from ILCK 7 subfamily. So among these, big one is the most extensively studied. So big one possesses a methylation motif. Actually here, it can facilitate its localization to the plasma membrane. And in the absence of flagellin-derived peptide FLAG22, big one is associated with both FLS2 and big one in an inactive state. Upon FLAG22 bind to FLS2, this interaction promotes the association between FLS2 and big one, and leading to a series of phosphorylation events, and the activated big one can further phosphorylate big one, which subsequently transphosphorylates the FLS2 big one complex. So this phosphorylation cascade occurs rapidly within 1 minute of activation. And follow this phosphorylation, big one undergoes monoubiquitination mediated by the E3 ligase RH-A3A and B, resulting in its dissociation from the FLS2 big one complex. So this dissociation is often coupled with endocytosis and then trigger downstream defense responses. So this process typically takes place between like 10 to 20 minutes after the ligand binding. I'm sorry. So downstream of RLCK, virus signaling pathways are activated, including those involving MAPKs, NADPH, oxidase that produce ROS signal, and calcium ion channels. And recently, a kinase known as DGK5 has been identified as playing a unique role in NADPH oxidation, RBOHD-mediated ROS production. So DGK5 is involved in the synthesis of phosphatidic acid, which is known as PA. PA can activate NADPH oxidase, particularly RBOHD. So multiple elicitors could induce dynamic phosphorylation of DGK5, as trigger both the PAMP from different pathogens and also some depth signals. And when analyzed the DGK5 after FLAG22 treatment with this post-tag gel, this gel could efficiently separate the phosphorylase bands. So after that, 2 distinct bands of DGK5 are observed here. So here we designated the lower and the upper migration band of phosphorylated DGK5 as the DGK5L and DGK5U, respectively. So this phosphorylation was further confirmed using the lambda phosphatase here. You see, after the treatment of this phosphatase, both these 2 bands are gone, right? So while both big one and I'm sorry here. So in big one or MPK4 mutant plants, these bands shift just disappeared. So which means these 2 bands are phosphorylated from DGK5 and MPK4. Although this both these kinase can phosphorylate DGK5, so the efforts actually are opposite. So big one phosphorylates DGK5 at serine 506, but the MPK4 can phosphorylate DGK5 at threonine 446. But big one phosphorylation DGK5 leads to a rapid burst of PA and activation of plant immunity, and encounters the MPK4 phosphorylation of DGK5 subsequently suppress DGK5 activity and PA production, and resulting in annotated plant immunity. So this interplay between big one and MP4 is crucial in regulating the PA homeostasis and the overall immune response of plants. So upon recognition of elicitors, so plants initiate a cascade of early immune responses, including a cytosol efflux of calcium ions, a burst of the ROS, and stomata occlusion to prevent further pathogen entry and deposition of kalos at the cell wall to strengthen the physical barriers. So these initial responses are followed by the late stage immune responses that enhance the plant's overall defense system. But notably, some early responses occur within just seconds to minutes, such as phosphorylation and calcium efflux rose burst, while others can last hours to days, such as stomata occlusion or kalos deposition, or even the growth inhibition. And some of the responses are transient, while some others are long-lasting. For example, the ethylene production, which can last for 1 day, and in cars 2 days, the calcium influx typically just lasts for half an hour. So pathogen infection for the infection in the plant and cause the perception of PAMPs by PR triggered PTI, right? So this will further lead to the transcription of some phytokine genes. So these phytokines undergoes maturation through cleavage or post-translational modifications and then secreted into the apoplast. So these phytokines are also known as the danger-associated molecular patterns. So these patterns are plant-derived small secreted peptides with immune modulatory functions, and this peptide functionally analogs to the cytokines in animals. So upon recognition of PRR, phytocytokine can induce danger-triggered immunity, which is also known as DTI. So similar to cytokines in mammals, the phytocytokines may act in autocrine or paracrine or and endocrine manners to activate or moderate immune responses. So the discovery of phytocytokines began with isolation of the cysteamine peptide from tomato leaves in 1991. So cysteamine is a 18 amino acid plant peptide. Hormone plays a crucial role in wood response. This peptide is identified in the Solanaceae family. So cysteamine induces the production of protease inhibitors, which serve as a defense against insect herbivores. So the receptor for cysteamine named SYR1 was identified in 2018, and in 2006, another endogenous peptide signal known as PEP1 was discovered in Arabidopsis plants. So this 23 amino acid polypeptides could activate innate immune response. And the subsequent research also revealed the PEPR1 is receptor. And today I will introduce you to two representative phytochemistries. So the first one is called SCOOP. So this peptide contains an SXS signature motif that is essential for its function, enable its recognition and binding to its receptor MIG2. Just like PAPs in PTI, the SCOOPs can also trigger various responses in plant immune response, such as MPK activation, actually here. What's really interesting is the SCOOP-like sequence are also found in a wide range of pathogens, including the fungal species such as Fusarium. In Fusarium, the SCOOP-like peptide is located at the N terminal of, just as shown here, of a highly conserved protein with unknown function. However, just like plant-derived SCOOP response in plant, this SCOOP-like peptide can also activate the MPK pathways. And notably here, why SCOOP-like peptides are widely distributed in fungi and bacterial species. The plant SCOOPs are exclusively found in brassica plants, such as Arbutus. So this suggests that the plant SCOOPs may have evolved later than their microbial counterparts. So therefore, the plant SCOOPs may be convergently evolved to mimic microbial SCOOP-like peptide and thus amplifying the SCOOP-like trigger immunity. And the second phytocytokine I want to share with you is the SCOOP peptide family. So SCOOP contain an N-terminal signal peptides domain, variable region, and also a C-terminal conserved domain with 2 conserved cysteine residues. So the expression of these SCOOP peptides is induced upon the perception of PAMs such as FLAP22, and the SCOOPs function as immune modulatory phytocytokines and significantly contribute to plant resistance against virus pathogens. So the SCOOP receptor, which named NOT, is essential for SCOOP-induced immune response. And the BIC1 again serve as the co-receptor here to regulate the downstream immune responses. And a unique feature of the SCOOP peptide here is its ability to suppress FLAP22 and abscisic acid-induced stomata closure. Because we know upon sensing initial infections, plants could respond to PAMs and the phytocytokines, leading to the rapid stomata closure to restrict pathogen entry. This process was known as the stomata immunity. And in responses, pathogens have involved the counter defense strategies, such as releasing some toxins or effectors to keep stomata open and facilitate the entry. And moreover, PEP-induced stomata closure creates a watery apoplast that promote the bacterial multiplication, and the prolonged stomata closure can also reduce transposition rates and negatively impact plant productivity. So to counteract these effects, the plants could induce specific phytocytokines such as SCOOPs to reopen the stomata. And this mechanism is termed apoplastic immunity. And this immunity can help reduce the water potentials and by disrupting the pathogen favorable watery environment necessary for pathogen growth and lesion development. So as previously mentioned, the co-receptor BIC1 is a critical component of the PR complex, as many PR interact with BIC1 to transduce signal into the cell. So apoptosis BIC1 is part of the circ family, which comprises 5 members here from 1 to 5. So PR could recruit BIC1 and SIRC4 to trigger a cascade of defense response upon detecting the PAMs from bacteria, oomycete, fungi, and dam peptides. However, BIC1 is also an ideal target for the pathogens to attack, right? And indeed, many bacterial effectors, including AVR-PTO, AVR-PTO-B, and HOP-F2 and HOP-B1, have been reported to disrupt BIC1's function here. So by this strategy can effectively, could efficiently interfere with the multiple PRR signal pathway, right? So this strategy of targeting BIC1 to simultaneously impair virus immune response is referred to in Chinese as kill two birds with one stone. And in 2007, two studies reported that the deletion of BEC1 and SIRK4 could lead to cell death in apoptosis. And this type of cell death is independent of the glycosylated and can result in necrotic spots on leaves. And this observation suggests a dual function for the BEC1 SIRK4 family. So on one hand, it can activate PTI, and on the other hand, it likely suppresses some certain forms of backup immunities. So BEC1, when BEC1 loses its integrity, the backup immunity function just works like a building standby generator, right? And activating compensatory immune responses. So this also raises the question, where does the backup immunity come from? And to investigate this, our lab utilized an efficient virus-induced gene silencing tools known as VIGS here. This is a system is used to create transient RNA interference in apoptosis. So the RNA targeting BEC1 and SIRK4 phenocopied the BEC1 SIRK4 mutant, exhibiting a cell death phenotype. So using this powerful tool, we can screen sequence tagged mutant library and identify several back to life mutants, such as the BTL protein, which involved in protein N glycosylation, and the BTL1 protein was found to work as a cyclic nucleotide gated channel, and the BTL2 function as a leucine-rich repeat RK. And notably, BTL2 can compensate for compromised PTI by activating phytocytokine-mediated backup immunity. So how does BTL2 work? I shared a model with you here. So this image illustrated BTL2 can collaborate with CNG20 to mediate backup immunity when BEC1 is disrupted. As I showed here, in normal condition, BEC1 can work as the co-receptor PTI and suppress the backup immunity caused by BTL2 and the CNG20. However, when BEC1 is perturbed, the phosphorylation of BTL2 by BEC1 is gone. So this was resulting in BTL2 activation, and this in turn could lead to the phosphorylation and activation of CNG20 and 19, and this will cause activation of robust backup immunity and finally even cause cell death. So for a long time, plant immunity was considered as a two-tier system, right? And when PR located on the cell membrane trigger PTI and the intracellular NLR-mediated effector triggered immunity, also known as ETI. So PTI and ETI share several overlapping downstream outputs, including the RS production and the cathode ion influx and the MAPK activation. And the transcriptional programming is also changed. Is also the convergent point. So recently, substantial advancement have been made in understanding how PTI and ETI interact to create a robust immune response. For example, the ROS is produced in both PTI and ETI pathway. PTI could typically trigger a rapid and transient ROS burst, but PTI and ETI together can induce biphasic ROS response. This is a pattern with a second stronger and more sustained burst. So this integrated perspective represents the plant immune system as a network with two mutually dependent components. So on one hand, the ETI can enhance the PTI response, for instance, upregulating the PTI components such as RBOHD, while the PTI can also can upregulate the expression of some NAR genes. So to sum up, when a pathogen invades, the PTI can act as the primary defense mechanism against the infection. So while the ETI is activated through the recognition of pathogen effectors. And the big one serves as a essential co-receptor for multiple PRR that initiate PTI. And the pathogen-induced PTI can stimulate the production of DAMs and phytocytokines, which further elicit DTI. And the DTI has the potential to amplify PTI and activate ETI. So this established robust protection against pathogen infections. However, the overactivation of ETI can lead to autoimmunity. So BAKE1 also play a crucial role in keep some components like BTR2 inactivated, and otherwise BTR2 may overactivate the DTI and the ETI trigger overactivated immunity. So BAKE1 here process dual role in activating PTI while also restraining DTI and ETI. And the BTR2 mediated DTI could compensate for disrupted PTI when BAKE1 is targeted by pathogens. However, in this area, there are many questions are waiting to be solved. I listed several future perspectives here. So first one is identifications of some new PRs and ligands, such as how many RKs and RP could serve as sensors for pathogen or dangerous threats, and how PR coordinate different PR and DTI signaling. And the second direction is the initiation and the dissemination of DTI, about how the phytocytokines are processed for maturation and translocation, and also how does these phytocytokines function, whether they are functioning in autocrines, paracrines, and endocrine signaling, and also the role of PR in PTI ETI integration are also going to be solved, like how do PR and the signaling regulate PTI ETI potentiation, and whether PRs can act antagonistically in PTI ETI interplay. And finally, it's still not very clear about the specificity in PR-mediated signaling. So whether there is any kinds of signaling specificity of different PR as a single cell resolution, and how does the shared co-receptors maintain the signal specificity? All these kind of questions are waiting for contributions in this area. And finally, I would like to acknowledge the many researchers and authors whose important and insightful work is not discussed here due to time limitations, and their contributions have been invaluably for shaping this field. And I also would like to thank my lab members for the invaluable assistance and guidance to preparing this talk, and my supervisors, Dr. Ping He and Dr. Li Boshan, had also been a tremendous support and engaging with me on the progress of this research area. And I'm also grateful to the University of Michigan for providing us a fantastic environment to conduct our research. I also like to extend my thanks to Dr. Harris, Karen, and everyone at MPMI for organizing this virtual seminar and providing me this opportunity to be here and share these topics with all of you. Thank you all for your support.
Thank you, Yingpeng, for an excellent seminar. That was really interesting, brought up lots of great ideas, so many directions from here. I can see lots of people virtually clapping for you, so that definitely reflects how I think we're all feeling. If people want to ask questions, please type them in the Q&A box on the bottom. That's where I'll be reading the questions from. So we'll get started there. One of the things I loved that you brought up, I just want to highlight this before we move on to the questions, is in your final questions, you were talking about this importance of single cell resolution. Are they acting in that cell? What about the next cell? How is this signaling work? We think about it often at a single cell level, but of course the plant is made up of many cells and many tissue layers, and it's just, I think, a really exciting and interesting direction. Okay, first question is from Ryan Kessens. Which components of PTI do you think have the greatest potential for editing and engineering to enhance and increase the spectrum of disease resistance?
Yeah, that's a really interesting question to, further apply our research on the daily and the agriculture productive, right? And in my point of view, the most greatest potential for editing or engineer here in PTI is some common components in this system, right? Just as I mentioned, like some co-receptors, like big-1, or also some kinds of IRCKs, they are shared components of this for many kinds of pathways from PTI. So maybe in the future, we can edit this component to make it a little bit more high efficiency to or trigger the more robust immune response. And I think this will enhance and increase the spectrum of the resistance. And not only in one kind of crops, but have a more broad spectrum for many kinds of crops. Yeah. Very interesting question. Yeah.
Yes, thank you. So I think maybe some people type questions into the chat, so just a reminder to type them into the Q&A and we'll read them. One thing that I also often wonder is thinking about this question when we think about the microbiome as being very complex, or thinking about this as much more nuanced than it used to be when they were good guys and bad guys only. And we now know that interactions with microbes are important in the life of the plant, not just in terms of killing them or sucking out their sugar. And so how, this is so interesting when we think about, since I'm coming from a symbiosis angle, it's intriguing because the plant is manipulating and using these to allow entry or interactions with some microbes, but not others. So given that last question about target for engineering, what do you think about it within the context of potential beneficial interactions?
Yeah, that's a very brilliant question. So actually, when I first joined this field, I also have the same question, right? So how the PR can differentiate the single unit from bad or good bacteria, such as the FLS2, right, is very specific to binding the FLAG22 peptide. However, many kinds of bacteria can have the flagella and then have this FLAG22 peptide here. So I think to answer this question, a recent publication already answered this very well. This is from I think from Jeff Dangle's group, and it told us actually we would better to answer this question from the perspective of evolution. Actually, for this what we call the good bacteria, they already evolved its own like the FLAG22 unit cannot be recognized by this substantial PR. This is magic, right? The bad pathogen can still keep this kind of PR, and plant PR plant PR can recognize these patterns from the bad pathogen, but not the good pathogens, and this will benefit the plant, like just as you said, take some benefits from the good pathogens, but keep these bad pathogens away from itself. Yeah, this is a very interesting topic. Oh, I'm sorry, Dr. Harris, I think you just muted yourself.
I did.
Oh,
Thank
yeah.
you. So we now have another question from the audience.
Okay.
JL Tan, who asks, how do you think the signaling would work in a combination of invasion in plants? For example, in the case where the insect vector brings a virus. Oh, yes, because some plant virus is noted to have some mutualistic interaction with the vector. And I think if we can understand that, we can help reduce plant virus in agricultural crops. It's definitely a very nuanced question. So this idea where some plant viruses do have a benefit, some promote things like thermotolerance. So, but on the other hand, not always good. So that's a complicated question.
Yeah, I think so. This comes to question because actually, about the virus infection in plant, I think not many researchers focus on the PR plays a role in this recommendation of virus patterns here. But about the signaling working in a combination of the invasion in plants, yeah, I think just like the plant, yeah, indeed it can face many kinds of stress at the same time, right? Not only from the abiotic, but also the biotic stress. But one interesting thing is, looks like many kind of these stimulators can cause a common downstream cascade. And but a more interesting question, but not unsolved, is it seems like some certain elicitors can cause a certain, the unique pattern of plants employed to fade off the pathogen infection, such as the screw peptide, as I just presented. It can reopen the stomata and let the water evaporated. Yeah, I think this could be a good example for answer the question whether the insect the insect together with the virus here. Although this, so could be a possible that although the insect and the virus can infect the plant at the same time, but the common downstream cascade, major downstream signaling is similar. But some certain species of insect or virus can also cause some specific responses, just as the screw did. Yeah, this is a very open question. We need to definitely much more efforts to answer this question. Yeah.
Yes, that's definitely a complicated one. We're moving on now to Aman Jayakar, who is curious about what is known about the role of PPRR in phytoplasma infection.
I think the question want to know the role of PRR, right? PRR and the phytoplasma infection. Let me see. I think the PR is regarded as the first line of the plants to recognize the infection of different pathogens.
Phytoplasma is very tricky, isn't it? The way it
infects.
Yeah,
it's very tricky. People don't have much study about these pathogens. This is a very interesting topic, and I think people today generally use some crops or apoptosis as a model plants to study the function of PR in plant innate immunity, and yeah, this
Yeah, no,
could
there's
be
some
a quite
interesting
open, interesting. Yeah,
research going on in phytoplasma, but there's still so much unknown that these
yeah, so much I know
connections
here.
are being drawn. Yeah, it's an interesting area of research.
Yeah,
Um,
yeah. Some PRR are conserved and limited to some certain species. Um, Although sometimes you cannot find a PRR in ptosis, but it doesn't mean you cannot find it in other plant species. Could be this PRR or some PRRs also functions in some certain plants and some or even some certain pathogen species. Yeah, but you just
mhm.
need to work it out to find a way to recognize it. And yes, this
Yeah.
is definitely one of the future perspective we need to explore more.
So another audience member asks, why does overexpression of some PRRs cause cell death when expressed transiently? Is it considered a hypersensitive response or a PTI response?
I would say, actually, when we talk about the status, we generally thought it's a contribution from the downstream ETI response rather than PTI. Because PTI is induced very quick, but relatively a little bit much weaker than the ETI. I think like some PR, indeed we found the reports some PR or even perceiving some elicitor can directly activate ETI, can directly induce the status caused by hypersensitive responses. But the mechanism is still unknown now. So yeah, this is also a interesting topic for future to explore. But
Yes,
in all, I
and
think
it's
this is definitely a hypersensitive response, but not a
yeah, and it's so interesting because when I first started learning about this, we had these very distinct buckets that we put things in. Is it this or this? And they were different. And I think now with what you were talking about with this much more integrated view of ETI and PTI as just a network of signaling, some of which are recognizing things outside the cell and some inside the cell, kind of brings that back to the fact that these are not as distinct as all parts of this same network.
yeah.
Okay, one more says, thank you for this lovely talk. I was wondering if
Thank you.
plants are able to differentiate between pathogen-induced damage and mechanical or physical damage when sensing damps.
Yeah. Yeah, that is a good question. So to directly answer your question, so if the plant are able to differentiate between the PAMs and them, I would say the extracellular domain of certain PR already did this work, right? But the noted what notable here is the PAMs and DAMs in the current understanding may occur in different time point, right? The current concept introduced that DAMs could be introduced by the recognition of some PAPs like FLAG22. It can just take the example I just presented here, like school, and the expression and the maturation of some phytocytokines can be induced by PAM, by PAM. Yes. So I would say there are many kinds of way for plants to differentiate the PAM and DAMP, both in different time point and by different receptors. Yeah, this is the smart point of this plant immunity.
Yeah, and this is a theme that you brought up before when asking the question, if
Yeah,
all these different specific receptors use BAC1, where do you get your specificity, or do you need the specificity?
yeah,
And it's a similar kind of question. Okay, another attendee asks, what do you think about the trade-off between resistance and growth? And what kind of resistance would be durable, right? Because you don't want to lose, right? If your whole goal is crop yield, you don't want to lose too much growth. On the other hand, right, you need some resistance. So what kind of resistance would be durable?
yeah. I think this is a meaningful question. And in my point of view, the plant facing the very challenging environment, not only from the abiotic stress and also biotic stress, right? So in this case, the plant have to respond to certain stimulus very quick. So that means if plant use the energy to be defense of the invasion of pathogens, it must be affect in most cases, the negatively affects growth of plants. So I think the resistance for the resistance of plant, it's better to have a way to just slightly activate the downstream robust immune response, such as ETI, but find a way just find the trade-off point. I mean the balance point to just slightly activate the immune response of the plant, but not too robust to cause any growth defect. Otherwise, it will affect the yield of the crops, right? So yeah, this is I think this is a future application direction of this for understanding of plant immunity. Oh, Dr. Harry.
So, yeah, I'm back. But I was just agreeing with you on that one. Definitely a direction. So our next question is from Iris Molhoff, who asks, how does BAC1 find all of these different PRRs in the membrane? Is there just an excess of BAC1 in the plasma membrane? Does every PRR just have a BAC1 nearby at all times?
Yeah, that is quite a detailed question. Yeah, what I didn't mention here, actually, if you notice, like many family members from this third family they are LRRK, but they have very short LRR domain. Like in BAC1, it can only contain, I remember, 5 LRR domain, right? But if you compare with some specific PRR like FLAG22, it has 28 LRR domains, super long domain, extracellular domain, right? So I think this is the reason why BAC1 can find so many different PR, because it's very short. So it can be more easier to bind with other PR because it has a simple structure, right? So your next question is
are they co-located within the membrane?
At all time. Yeah, it's not necessary to for PR always have BAC1 nearby, and also not all PR need the co-receptor of BAC1, right? But what I can tell you is the interaction between PR and BAC1 is very, very strong. So I think, yeah, in the normal condition, many PR could have the BAC1 interacted together, but the key factor here interacts the activation of this complex is the binding of elicitor and the downstream phosphorylation of this PR and the BAC1, right? So you can just imagine these two proteins just get together, but it's always done by once it can perceive some elicitors, it can quick phosphorylate it and activate downstream like RFCK and cause plant immunity. Yeah, this is a clever strategy.
Yes, there's this also reminds me of like non-factor signaling and symbiosis, where there's also a co-receptor. There's two halves of receptor co-receptor, and they are associated with certain microdomains in the plasma membrane, right? So remorins, flotillins, and there's this sense of a microdomain which may reorganize some of the components of a signaling complex. So when it binds, it's ready to go. So there may be other, it's possible that some of these are floating around in the membrane and just waiting, and it's possible that others are reorganized, maybe in response to prior life history or maybe just something about the kind of plant. So you may even get a mixture of that, of some that are kind of waiting with the other components nearby to signal, or some that just have to find them. Maybe that's some of the timing of some of these different responses. So our next question is from Aurélien Boisson-Dernier, who asks, which one came first, plant peptides to regulate immunity or pathogen peptides to manipulate plant physiology? This is a chicken or the egg question. And are they examples of horizontal gene transfer, or is it mostly convergent evolution? It's a very good question.
Yeah, very good. This is a very good question. Like just like in the example I just listed your scoop, this case it looks like the plant evolved like slower than the pathogens, right? But what I can tell you is there are also other kinds of peptide in plant, but also from the signature in pathogens like the rough peptide, right? But in their case, it seems like the plant evolved quicker than the pathogens did. So for answer your question, I would like to say both. Both the horizontal gene transfer and the convergent evolution happened here in the interaction between pathogens and the plant receptor, but case by case. And for specifics, we need to do more research on the evolution perspective to see whether what really happened there. Yeah, that's a very brilliant question.
Yeah. Okay, we're only going to answer 2 more questions and then we'll finish. Lots of great discussion here. Our next one is from Chandan Moria, who asks, you mentioned the role of BAC1 as a co-receptor in various immune responses. How does BAC1 specifically contribute to immunity against root-related pathogens? And what challenges exist in studying BAC1-mediated defense in root tissue compared to above-ground plant parts? Well, that's always the tricky part about roots, right? They're below ground in the soil, but they can be studied. But that's a good question. How does BAC1 contribute to immunity?
Yeah. As I just said, this is also a magic part, right? Like some PEP, like PEP1, like some PEP, like phytocytokines such as PEP1 or scoops. They also have the relies on the co-receptor BAC1 to cause downstream responses, right? But these 2 peptides, PEP1 and the scoop, they have very significant effects on the inhibition of root growth, right? But if you compare with other elicitor like FLT2, although it can affect, but it's not very significant. Yeah, this is also a tricky part. I think this is also the challenge point here. So how can we tell where did this specificity of the co-receptor came from, right? All these kind of PR all go through this convergent point, but it finally gives rise to totally different responses. Yeah, right. And your question 2 is
what challenges exist
in root tissue compared to the above ground plant parts? I would say I didn't do much research on the root tissues, but the challenge if from technically speaking, challenge would be root tissues. You need to do quite a lot of microscope work, which is very time consuming and maybe not very easy like people did the immunity on the above ground plant parts. But I think it's if necessary, like for some elicitors, you already can see the significant change in root parts. It's still worthwhile to explore what happened there. Yeah, and I know people already developed quite some cutting-edge technologies, studies the root. I mean, to observe the growth of a root, the direction, and different time point to observe root with different types of elicitors, such as the dwarf. Yeah, this would be very good point to explore in the future.
Yes, so we are going to have one more question, but I just want to remind people that they can go get more detail out of this by reading the article in the MPMI journal. It's connected, it's linked to where you did your registration, and we will post it in the chat. I think it was posted earlier, but here we have it, posted in the chat, so you can link to that. And remember that there is a person to contact with email questions for every article. Sometimes there's two, and that's a great way to continue this conversation. And I'm sure Yingpeng would also be interested in discussing this with people, either by email or at some next conference. And also wanted to remind people that this HH4 review is an exciting series of reviews covering key topics in plant microbe interactions. And this is the first one we've highlighted in What's New in MPMI. But if you go to the MPMI journal website, you'll be able to find the list of other reviews. And if you enjoyed this, you may really find the others to be useful and interesting. Now we're going to move on to our last question by Laurence Binschedler. Are the PRR in microdomains, raft-like detergent insoluble membranes? This was kind of what we were hinting at before. So what's the organization within the plasma membrane? What's known?
Yes, my answer is yes. The PRR, indeed, you can find it in the microdomain on the cell membrane, and it also floating on the cell membrane like the raft. So what happened here in my mind, if you have some experience in observing the microscope like turf, you can see the surface of your PRs on the cell membrane. So you can see many dots, just maybe evenly or unevenly distributed across the whole cell membrane. So, but the cool thing is this point represents to the individual PRR molecules. They are dynamic. They're not completely steady there. They keep moving very fast, or sometimes it can even gathering together and to a certain part and undergoes endocytosis to exert the downstream signaling. This is about the dynamic of these behaviors of PRR on the cell membrane.
Yes, interesting. So many different areas to go from here. So thanks everybody for your participation. Thanks especially to Yingpeng Xie for this wonderful seminar. Remember, this will be, thank you. This will be recorded, or was recorded, and so it will be posted hopefully in about a day or so. If anybody would like to help edit the transcript, that would be terrific, because that helps us to make it more accessible. If you're interested in participating in this way, please contact me by email, and I would be happy to work with you on that. So thanks again, Yingpeng. And
thank you, Dr. Harry.
You're
thank
welcome.
you, everyone.
Thanks everyone for your participation. Great discussion. And there are a couple of nice notes in the chat about how much people enjoyed the seminar. Okay, thanks everyone. Goodbye. Till next time.
Bye.